Good morning, everyone. Welcome to the 2020 PEP presentations. I'd like to invite George Lyles to welcome us and Joniqua Howard to also welcome us and tell us a little bit about how this is going to work in this virtual format.
So who goes first, me or
Good morning, everyone. Welcome again to the 2020 PEP Virtual Student Research Symposium. Before we get started with our introduction by George Lyles, our 2020 PEP Director, I wanted to let you all know that today's symposium is divided into two sessions. Our morning session from 9 to 11 and our afternoon session from 1 to 3, followed by our PEP graduation. All meeting participants have been placed on mute, and if you have any questions throughout the 15-minute presentations, please be sure to type your questions in our chat box. We will have approximately 3 to 5 minutes after each talk for questions if time permits. That will be read aloud. Without further ado, please welcome George Lyles.
Good morning, and thank you. Can you hear me? Yep, okay. First of all, I have to say that was an awesome video. We must have hired a production company to do that. I don't know, Joniqua or Onji, who did that, but that was awesome. Thank you. I do want to welcome everyone to the PEP Symposium. I want to welcome, of course, the students, the PEP staff, and the mentors, and all of the various friends of PEP who join us at this time of the year to hear from our students. I also want to just say a word about what a remarkable summer it has been. In March and April, when we had to decide whether or not we were going to do the program, and if so, how, it was a challenge, and it was a remarkable coming together of talent and passion. So the PEP staff, which is larger this year, just did a remarkable job, in my view, of building a program. Same content, different delivery is what we were aiming for. And I'm really looking forward to hearing this morning the results of that when we hear from the students. The thank yous that we need to acknowledge the 7 partner institutions, the 6 Woods Hole diversity institutions here in Woods Hole, and our academic partner, University of Maryland Eastern Shore, all of whom contribute substantially to make the program work. I also want to thank the PEP staff, which, as I mentioned, is large this year. Ben Harden, Louise Valentin, Andre Price, Kelly Luis, Joniqua Howard, our advisors, Ambrose Gerald, Onjalé Scott Price, Hauka Kite Powell, Paul Joyce. And I especially want to highlight a thank you to Onjalé Scott Price for stepping in this summer when I had to take some leave, and just running a really excellent program. So we were fortunate to have Onji here, available, willing, and able to lead us. So thank you for that, Onji. Oh, the mentors. I want to thank the mentors. We thank the mentors every year, and I ask the students every year, please say a thank you to your mentors. That has to be a double thank you this year, because back in March and April, we went to the mentors, many of whom are first-time PEP mentors, and we said to the science community, we need people who are willing to help us. We don't know how to do this, but we need people who are willing to help us try to figure out how we can provide mentoring and research experiences that are done at distance. So I think it was very brave of our mentors to step forward, and I think we really owe our mentors a big thank you. So with that, I'm going to go on mute and look forward to hearing some really good presentations.
Thank you, George. Our first presentation will be given by Ahiamadjie from Delaware State University, who's been working with Dr. Elena Peredo from MBL. He will be discussing identifying sulfate-reducing bacteria by assembling genomes from eastern U.S. salt marsh habitat. Without further ado, Ellie.
Hello and good morning, everyone. First off, I want to thank the PEP program and everyone who helped run the program for giving me the opportunity to actually experience real research for the first time and allowing me to begin my metamorphosis into being a true scientist. Again, I'm so thankful for the opportunity, and it truly changed my life, even though we had to stay home. But my project was about identifying sulfate-reducing bacteria by assembling genomes from the eastern US salt marsh habitats. My mentor on the project was the wonderful Dr. Elena Peredo, a renowned scientist from the Marine Biology Laboratory in Woods Hole, and my project partner was the distinguished Jamie Jaban. Throughout this summer, us and another group of scientists worked on collecting data from salt marshes in Alabama and Massachusetts so that we could better understand the microbial activity taking place in these salt marshes. Before I get into our actual research, I want to give you background information on what the areas we got our samples from are like. Salt marshes are coastal wetlands that are constantly affected by the movement of the tides. They are constantly flooded and drained with salt water throughout the day, hence the name salt marshes because of the salt water. The soil is usually composed of deep mud and peat. Peat is basically decomposing plant matter that is usually several feet thick. It's saturated with water, filled with roots, and it's really spongy. Salt marshes are important to us because they are an integral part of our economy and our culture. They provide essential food, refuge, or nursery habitats for more than 75% of fishery species like delicious shrimp, blue crabs, and finfish. They even protect the shoreline from erosion and reduce flooding. Because salt marshes are constantly submerged by tides and also contain a lot of decomposing plant matter, aka the peat that was previously mentioned, oxygen levels can be extremely low in these areas. This is mainly caused by the growth of bacteria that produce the sulfur smell that, if you've ever been in a marsh in person, will know smells like rotten eggs. So it's not appealing at all. But this bacterial activity was what my team wanted to explore and understand. We wanted to see what bacteria was present in the soil and deduce how a plant like Spartina alterniflora could possibly grow in an area like this. Before we get into the bacterial aspect of the project though, let's get to know the plant that's helped start it all. Spartina alterniflora, better known as cordgrass, is commonly found in salt marshes along the Atlantic coast of the US, the eastern side. Salt marsh cordgrass can grow to be 3 to 8 feet tall, so it's no small plant by any means. And they usually grow tallest towards the water and shortest away from the water. Spartina alterniflora is a major reason why salt marshes are home to a plethora of species. The roots and the base of the plants provide an excellent place for animals like mussels to collect and call home, and for other creatures to hide from predators like birds. And although it was previously thought that Spartina alterniflora was able to survive solely due to its ability to expel the salt from salt water through its leaves, scientists now understand that the bacterial activity taking place in the soil may also play a part in its survival. More specifically, the involvement of sulfate-reducing bacteria and sulfide-oxidizing bacteria. Throughout the project this summer, my focus was on sulfate reduction, or more specifically, what sulfate-reducing microorganisms were present in our salt marsh soil samples, and understanding what their role was in the salt marsh sulfur cycle. So what exactly are sulfate-reducing microorganisms? Sulfate-reducing microorganisms, or SRMs as I might refer to them throughout this presentation, are a very diverse group of microorganisms, mostly in the bacteria domain. The functional significance of SRM diversity is not yet known, meaning that scientists don't really understand why there's such a large array of sulfate-reducing organisms, but they do recognize that the diversity truly does exist. And these organisms are capable of creating a plethora of byproducts in the soil. And what exactly do these microorganisms do or use to survive? So sulfate penetrates deep into the soil because of its high concentration in the water in these areas. And they supply sulfate to the sulfate-reducing microorganisms. Their energy source is the buried organic matter, which is bountiful in salt marshes because thanks to the peat. And they oxidize the organic matter into CO2 and other inorganic nutrients. Sulfate is to sulfate-reducing microorganisms as oxygen is to us. Meaning sulfate is used for respiration by SRMs, comparable to how oxygen is used for the way humans respire, how we breathe it in. Same thing applies for sulfate-reducing microorganisms with sulfate. Although this all might sound like a really simple and straightforward process, the sulfate reduction process is actually really complicated. So sulfate is taken up from the environment by these sulfate-reducing microorganisms by sulfate transporters and become activated with ATP in the cytoplasm from the SRMs through the enzyme ATP sulfurylase. Anywhere that you see has A's at the end is usually an enzyme, which is being used by the sulfate-reducing microorganisms for various processes. Now, they use that SAT, that sulfurylase, to form adenosine 50-phosphosulfate, which is that APS. Now, the APS is reduced to sulfite by another enzyme, adenylyl sulfate reductase, which receives electrons from a membrane-bound electron transport complex, the ETC. Now, that bisulfate is further reduced to become hydrogen sulfide by the bisulfite reductase, that's the DSR complex. Now, this produced hydrogen sulfide later on diffuses passively through the cell membrane of the sulfate-reducing microorganisms into the environment, which will later be used by sulfide oxidizers, which my partner Jamie will be talking about later. So by using a comprehensive database on sulfate and methane in the seabed and using environmentally calibrated algorithms for geographic extrapolation, the scientist Egger et al. Was able to develop a map of sulfate reduction for the world. And that's what you see here. As you can see, there's a lot of activity taking place along the continental shelf around all these continents, and it decreases going out deeper towards the ocean. So what did my team exactly do for this project? I'm going to give you the diet version because my partner Jamie will really be going into depth and detail about the specific programs and processes that we use for our project. And I don't want to be repetitive, so I'm not going to go ahead and say all that now. But what our team basically did was first download reads from a data bank to be processed. These reads were, like I said, collected from the Alabama and the Massachusetts salt marsh areas. That's where we got our data from. And then once those reads were downloaded, we were able to use various programs to check the quality of our reads so that we can later on assemble our reads into contigs, which are basically DNA segments, which we can use to see what bacteria is present in the soil. And even if we know what type of bacteria it is, what their role is in the process that they're doing in the soil is actually. So after doing all that, I did actually get results. We were able to get results before the program finished, and I have a video right here going over the results taken from the Alabama marsh samples. I think the audio doesn't work very well, so I'm going to play the audio from my phone, but also show you the video from the laptop. This diagram is showing the percentage of the bacterium that was from our samples. Now, as you can see, 85% of the information that we collected is no hits, meaning that we do not know what that bacteria is. It's not been cultured and sampled yet for us to, or studied yet for us to be able to understand and know what type of organisms are present. But we do know 12% only 12% of the sample that we collected was information that we actually do know and hold now today. This is a vast array of bacteria, as you can see. Even so, 12% is a huge amount of information. While looking at sulfate-reducing bacteria, I specifically looked at the epsilon subdivisions, more specifically the sulfobacterials. Now, this is where I was able to find my
Ellie, we're not able to hear that very well.
Marshes. And this whole area only accounts for 0.9% of the full sample that we had taken from the area. And this is also about 7% of the bacteria that we were able to extract. And when you go more deeper into these sulfate-reducing bacteria, it gets less and less. It's only 0.3% of the whole data that we collected. And it's still a lot of information, as you can see. This accounts for about 3% of the bacteria in the samples and about 5% of the proteobacteria as a whole.
Yeah, so those were basically the findings that I had gotten from the Alabama marshes, and Jamie will be going more in depth about how we were able to get that diagram in the first place. So again, I want to thank everyone in the PEP program, especially my mentor, Dr. Elena Peredo, Dr. Gerald, everybody that was able to run the program, as well as the people funding the research that was taking place and giving us the opportunity to be able to actually be involved in this extravagant work. So thank you very much. Do you have any questions?
I am not seeing any questions here, so I'll move forward.
I do see one question, actually. Bill Waite says, awesome talk, Ellie. How deep do the Spartina roots go?
The Spartina roots, they go, depending on how tall the plant is, they go deeper depending on the height of the plant. So if the plant is super tall, then the roots will be going deeper. And if the plant is shorter, away from the water sources, then the roots will be shorter.
Thank you.
We also have one more question here from Sandy Sutherland. Please recap your results because the audio was a bit garbled.
Oh, okay, I'm sorry about that. I can just explain the results right here, and I'll fast forward through so basically, out of the entire sample that we got, 85% of it was not known bacteria. So it's not, so scientists do not have labels, and we've not cultured that bacteria to be able to identify them. But we do have about 12% of that bacteria. That's what is right here. And out of that 12%, I was specifically looking at the sulfobacteriates. Hold on. Yeah, right there where you see the where my cursor is. That was the sulfate-reducing organisms that I was specifically looking for. And then that was about, I believe, 3% of the entire sample. No, actually, it was about 0.9% of the entire sample, including the 85 that we do not know about. But this area was accounting for 3% of the bacteria that we actually did know and were able to find. And yeah, and that's what we were able to see, how much of the sulfate-reducing bacteria was present in the sample.
Thank you, Ellie. Moving on to our next presenter, you set the stage quite well, Ellie, for your colleague here. We will have next presenting Jamie Jabone from the University of Massachusetts, Boston, with his research mentor being Elena Peredo from MBL. His topic will be assembling metagenomes of sulfide oxidizing bacterial communities in eastern coastal Spartina salt marshes. Without further ado, Jamie.
Hello, everyone. Can you see my screen? Yes? All right. So My name is James Jabon. I am a research mentee for Dr. Elena L. Peredo, and my partner is Ahiamadjie, and he provided you guys um, with the like first dish of the course, and I will be providing you guys with the main course, the meats of the project. So my project is on assembling metagenomes of sulfide oxidizing bacteria communities in eastern coastal Spartina marshes. So the first part of this presentation will actually be a quick recap of everything Ellie talked about. And so the objectives of this project, right? It's to better characterize the genomic diversity of sulfur oxidizing bacteria in salt marshes. So the reason why sulfur oxidizing bacteria is important in these salt marshes is because sulfide in these marshes are actually quite toxic for these plants. They actually killed them. And these sulfide oxidizing bacteria exist to convert these sulfides into sulfates, which are actually nutrients these plants can uptake to consume. Now, a quick salt marsh overview. The reason why salt marshes are here and the reason why they exist is because they form a natural barrier between the harsh conditions of the ocean and, oh, can you guys hear me?
Yes.
Oh, okay, sorry. The harsh conditions of the ocean, and it prevents against the mainland. So the ocean will definitely will degrade the mainland if these salt marshes didn't exist. And the main organism we can see in these marshes are these Spartina grasses, these giant grasses. They're actually related to mainland hay, surprisingly. But that's not the focus of our project. We're more focused on what lies beneath the soil of these grasses. And these are sulfide oxidizing and reducing bacteria. What you can see in the soil, in this little red box, is this black layer of sediment. This is where all the sulfuric activity is taking place in these organisms, and that's our focus. This is where the sulfur cycle is happening. Ellie already explained what's happening with the sulfur cycle in the reduction part, but my interest is in the oxidation part, but I won't really be going into detail like he did. But the main takeaway is sulfide is oxidized into sulfate, and sulfate is reduced into sulfide. And that's what you need to take away from the sulfur cycle. And this stuff does occur naturally and geochemically, but microbial organisms speeds this up radically. So this is our project. Our project is a metagenomics project. And what are metagenomics? Metagenomics is a study of genetic material recovered from environmental samples. And these samples are then converted into data, which we can use to understand the community of microorganisms present in the area. And how did we do this? There are multiple steps that are involved. First thing we have to do is go study the area. After we study the area, we need to gather the samples. Once we collect the sample as desired, we have to extract the DNA from those samples, and then we need to sequence the DNA into data. Once we have this data, we have to analyze the data. But for our particular case, all we had to do was analyze the data. This data was taken from bioinformatic databases and from various projects, and each piece of data we took is called a run. And this run can be from MGRAS or NCBI, and they can range from a wide variety of sizes. But for our particular case, they were actually a whopping 4.5 gigabytes each, and we had a good number of them. So we did run into cases where we ran out of space in what we used to actually process the data. Before I go into that, we need to assemble the data. The best way we can describe what we did was build a puzzle, because we had to sequence this data, and then we had to assemble the data up like a puzzle piece to actually identify what we're looking at. And we use this by using the operating system Linux. Linux is an open source operating system, and you can create programs, modify programs, and share programs with everyone across the world, all for free. And that's what we used. We actually used this by connecting to a bunch of servers. And these servers are massive. They can be 10 to 100 times the computational power of regular computers, because that's the work that we're dealing with. And again, we had to collect this data, assemble them into pieces, and then reassemble them into, again, to create our genome. So we had, our data was initially reads, which are actually short nucleotides, 150 nucleotides long approximately, and then we need to piece them together to create contigs. These contigs are massive, 1,000 to 250,000 nucleotides large, and then we have to piece those contigs together to create a proxy genome, or approximate genome of an organism. And that's called a pipeline. Those are pieces of the pipeline. And in this pipeline, we had the raw reads, we generated the good reads, and by using a bunch of programs that filtered and trimmed our raw reads and took out all the bad sequences and only left the good sequences. And we did a de novo assembly, meaning we're trying to reassemble the genome of the target organisms. And then after we run this de novo assembly, we create contiguous sequences, or contigs. Then we run it through a program that would assemble these contigs, so we can then generate a taxonomy. So we worked with 3 separate projects. We worked with projects from Raleigh, Massachusetts, Alabama, and Plum Island, or IGERT data. I'll be referring to Plum Island as IGERT because that's the original name of the project. And in each project, we had 25 samples from Raleigh, 2 from Alabama, and 32 from IGERT. So imagine that. Imagine each of those samples 4.5 gigabytes large. That's a lot of processing power we need. And as you can see from the original number of reads, it starts from 1,000,000, 100,000, 200,000, and we had to trim it down to significantly smaller number of reads, especially for Raleigh, Massachusetts. And then we run a de novo assembly using MetaQuast, and that's our evaluation tool with the contigs. We're trying to see how well our contigs look and line up and compare them to each other. We did this 3 different ways. The first thing we did was we did a row of sequencing depth from IGERT. I'll go into more detail a little on. We did a selection of suitable data sets from Raleigh, and then we did a comparison of different assemblies using all the projects. So row sequencing depth from IGERT, we just wanted to see that we wanted to compare the contig sizes with our samples and compare with, like, sorry, my voice, and compare how the contig sizes actually relate to the cumulative length of our genome. The main takeaway from this graph is, as the size of your contigs increase, the cumulative length of your genome actually increases as well. But what's really interesting is you have these small fragments that are beneath the final contig, and those fragments, even though they have the same contig length as the final contig, which is the giant blue graph over here, if you can see my mouse, they generate actually a smaller cumulative length than the final contig, which is very interesting. And then our second part is selection of suitable data sets. So our Raleigh data set was absolutely massive. We needed to trim this down, or this would never be done on time. It would probably take us 2 weeks. So we trimmed it. So we decided to remove contigs that are less than 2,000 and 3,000 base pairs to see which one is better to work with. And as you can see from the number of contigs, just removing 2,000 or 3,000 base pairs, we either lose 75% of our contigs or 90% of our contigs, which is insane. But if you look at the GC content of the graph, the curves are actually very similar. So we didn't really lose that much information when we removed the really small contigs. And for our third comparison, we compared the different assemblies for all 3 locations, or all 3 projects. And what we find is something really interesting. We're looking at the GC curve. And for the Rowley curves, everything looks great. We have high peaks, but for the Eigert, we have this massive bump, and we are not sure why this bump exists, but it's really interesting, and we'll go into, and it's its own analysis of its own. Now, we're going to identify the organisms for each project. To do this, we need to run a taxonomy. So for taxonomy, we're looking at the taxonomy labels for these genomes that we created. And then we have to visualize this taxonomy using a program called Krona. So I'm going to play a quick video, but I'm going to talk over this video. As you can see, we're at the kingdom of bacteria because that's our kingdom of interest. And as we go further down this taxonomy, we go into protobacteria for their phylum, then gamma proteobacteria for their class, and we go into Therotrachilus for the order and Therotrichacea for the species. And right there, I highlighted my targeted organism, Begatoa leptometoformis. And the reason why I targeted this one specifically, I'll go into further detail in the next slide about that. Keep that in the back of your mind though. But these are all sulfur oxidizing bacteria in these sites. There's 546 sulfur oxidizing bacteria in Rowley, 508 in Alabama, and 515 in Eigert. And I didn't know how relevant this information was, but there's 402 taxa found across all the locations. That's super interesting because it means they have a lot in common, despite their geographic distances. And remember the bacteria Begatoa leptometoformis that I mentioned earlier? Well, the reason why I highlighted this one, I was looking for this one specifically, is because it is the most prominent sulfide oxidizing bacteria found in all the salt marshes. They're the biggest contributors, and this is how they look like. They form this massive filamentous layer in the marshes, in the water, and they are related to what we call cable bacterias. I'll go into further detail about cable bacterias at the end of this slide, but what the main takeaway from this is that the sulfur cycle is incredibly complex, and we need these bacteria because they are the ones that help the Spartina grasses maintain the structure of these salt marshes. They clean up the soil for them so they can grow and prosper and hold the front line against the harsh oceanic conditions. And in these yellow boxes is the complex cycle for sulfide oxidizers have to go through. And Begatoa, which is a very amazing bacteria, it's one of those cable bacteria. So they form these long chains that reach probably to 1 kilometer underneath the soil, and they actually uptake all of the sulfide and convert it into sulfate to clean up the whole area for Spartina salt marshes to actually exist. And my last comment for this slide is if you see these purple little balls in this image right here, you can see that this is bacteria. They're called purple sulfur bacteria. They're sulfide oxidizers as well, and they are really unique, and this is a fascinating project. I want to thank everyone in the Woods Hole community for helping me be part of this program. I don't know what I would have done this summer without you guys. This community is amazing, especially to my mentor, Elena Peredo. She was amazing with her team, and also my partner. He's my man, Ahiamadjie. Amazing guy. He comes in clutch all the time, and also the COVID-20 crew. I remember y'all. You guys are the best. And thank you so much for listening to my presentation. Any questions? Also, this is extra parts. So any questions, guys?
Thank you, Jamie. For the sake of time and to remain on schedule, we're going to ask that if you have questions to message him directly in our chat box. Thank you. So our next presentation will be visualizing invisible boundaries in the ocean distribution of water masses in the Northeast US shelf, presented by Ayanna Butler from Howard University, and her research mentor is Dr. Stace Bolu. Without further ado, I present to you Ayanna Butler.
I'm sorry, I was on mute. I apologize. Okay, so good morning, everyone. Thank you, Ellie and Jamie, for such great presentations. So again, this is my presentation on visualizing invisible boundaries in the ocean, distribution of water masses along the Northeast US shelf. And so this data was done by the NES LTER, which is the Northeast US Shelf Long-Term Ecological Research. So this project was just recently funded. And so looking into water masses is a new topic for them. So to be able to tackle this project was something very exciting. And so what actually is a water mass? A water mass is a pocket of water with its own unique qualities. And so you can see kind of in this diagram at the bottom where this deep blue is, it's called the cold pool. This will be referred to as shelf water in our presentation. And so it has, it's kind of like a pocket. It has its own set of temperature and salinity, which is what we'll be looking at in this presentation. And then you can kind of see these, I guess you would say, rainbow looking colors, green through orange, which would be our transition water. And then this peach kind of color is our slope water. And you can see it's called these things because this brown mass, which is the land, is the US continental shelf. So as you travel down the shelf, you have your different water masses. And so it's important to even look into these water masses because they influence biodiversity on the shelf. So the water masses move throughout the year and as the seasons change. So if a specific organism likes a certain temperature and salinity, like in the cold pool, if the cold pool has moved, then you won't see that species where it was originally. So the goal of this presentation is to create a workflow that will determine these water masses. And so the workflow we're going to be specifically looking at is chunks of code that also have descriptions in them so that if anyone wanted to just click on the link, someone who knows nothing about science, they could read the descriptions, look at the chunk of code and the output from the code and kind of interpret what's going on and what the goal of even writing the script is. And so again, the goal of this project is to create workflows that identify these water masses. And again, the data will be coming from NES LTER cruises. And these images just give you a better idea of where the Northeast US shelf is. So we're kind of looking right in the Cape Cod, end of Long Island region. And so again, the purpose of this project is to produce an R script with flexibility for other people to use so they can identify water masses from their data. So essentially in the script that we created, there's just one specific line that lets you read in the data. Oh, excuse me. There's one specific line that lets you read in the data. And the goal is that anyone could take whatever data they're using and input it in that line and be able to get the same results, well, based on their data that we got in this plot. So that kind of tackles the idea of reproducibility, which is a huge concept in science because we all want our work to be able to be done by someone else because that just proves the validity of what we were doing. And so another thing that we're able to do this year was compare our 2019 and 2020 cruise data. So the workflow that we created was based off 2019 summer data from last summer. And so this summer, actually about last week, we had a 2020 cruise go out. And we were able to get the data from that cruise and input it into our script and get results. So that was very exciting. So we'll be able to go look at that. And then lastly, the script was posted on a site called GitHub, which lets you share scripts and use version control so that other people can look at what you're working on. So some keywords, again, that you'll be hearing throughout this presentation are shelf water, transition water, slope water, and Gulf Stream influence. And again, this diagram just kind of goes through what we went over before about where the shelf water is, where the transition water is, and where the slope water is. Gulf Stream influence really doesn't affect this presentation, but we'll be able to see that our shelf water is closer to where land is, and it's going to have a lower temperature and salinity, and then we'll transition, and then we'll have our slope water, which will be a more deeper ocean, which will have a higher temperature and salinity. Okay, so the first thing we needed to do was create a temperature and salinity plot. And I want to give a huge shout out to Dr. Ben Harden, who was our PEP course. He gave our course this summer. And so I was able to work with him and adapt from his script about how he made a temperature and salinity plot for a paper that he was working on. So what a temperature and salinity plot is as the cruise goes along the shelf, it picks up temperature and salinity at specific locations. And so we're able to track the temperature and salinity along the shelf. And if you can see in Dr. Ben Harden's plot here to the right, he has multiple cruises, and we're just working with one cruise. And his is contoured by depth. And so you can see this cluster in this bottom left corner is the cold pool, and this cluster up in the top right corner is the slope water. And this is based on the temperature and salinity readings that he got. So he was able to set limits just based on previous knowledge and from this data to be able to determine what the temperature and salinity limits would be for the cold pool and for the slope water. And so this is something that we needed to do in our project as well, is use previous knowledge and knowledge from the plot that we got to be able to set the limits for temperature and salinity. And so when we do our plot, our contours will be done by latitude, but essentially we'll have kind of one line of what Dr. Harden has in his plot. Okay, so this was the temperature and salinity plot that we were able to produce in R with our data. And so as you can see, our cold pool or shelf water is kind of this big rectangle here, and it ranges from about 30 to 32 in salinity and about 19 to 25 in temperature. And you can see this top right box is our shelf water, and it kind of, slope water, and it kind of ranges from about 34 to 35 in salinity and 26 to 27-ish in temperature. And so if you pay attention to the sizes of these boxes, they'll matter a little bit later in the presentation, but this was the readings that we got for temperature and salinity along the shelf for the 2019 cruise data. And so the next thing we need to do was actually locate these water masses on the map. So we again took a script that we got from the PEP course in order to be able to create this map. And we're gonna use the limits that we got from the temperature and salinity plot in order to locate these water masses along the shelf. So this was the map that we were able to produce in R. You can see we're in the Cape Cod region. And like I said, when we paid attention to the boxes from the temperature and salinity plot, you can see how the shelf water kind of had a big, long rectangular box. This is what you would see for the location of it along the shelf. And you could see the bluish, greenish color for the shelf water is in reference to the same size as the box. And then you have our transition water, which is the peach color. And then you have our slope water, which is the purple color. And that also references the box that we saw on the temperature and salinity plot. And so we were able to identify where these different water masses are along the shelf. So will this notebook actually work for the 2020 cruise? I kind of gave that away, but it did work for the 2020 cruise, which is extremely exciting because 2020, like, it just went out last year. The cruise just went out last week, excuse me. So to be able to get that data and have our script work on one try to be able to interpret the data was extremely exciting. And 2020, as we all know, is just a different time as opposed to previous years. And you can see cruises are handling things differently. You can see everyone is social distancing and everything is just kind of different about 2020. So to be able to get results from 2020 was extremely promising and exciting. So again, we have our 2019 temperature and salinity plot. And then we have our 2020 temperature and salinity plot. So you can see the limits for the shelf water are a bit different. And then definitely the limits for the slope water are extremely different for this year compared to last year. And part of that is because we did set our own limits. So there's no like hard and fast limits on where shelf and slope water will be. It's kind of just all based on what we know. but you can see that there is a difference, but we were able to get a plot, and that's the main goal of the script. And then again, we have our 2019 water mass location map, and this is what we got for 2020. And so some differences that you can notice is the location of the slope water. There's a lot less slope water in the 2020 readings that we got. And also something that my mentor and I noticed was that the shelf water ends around at the same latitude, which is typically doesn't really happen that much because the water masses are always moving, but we found it extremely interesting that they were in the same location as last year. And so our final results from the workflow, again, our workflow was posted on GitHub. So this will let you see, again, the descriptions and the code, and hopefully everything will make sense as we go through it. So again, this is the title, Our Notebook for Water Masses in NES LTER Underway Data. And then so you go in, you would load your libraries, create your contour plot, set your limits for your shelf and slope water, what you think it will be, and actually plot your temperature and salinity plot. And then as you would go through, you would categorize your water masses, and then you would set some latitude and longitude limits, and then you would plot your water mass location map. And then this workflow also goes in the next steps because it's important to know that research is never done. There's always more things to look at. So as a end user looking at this script, you'll be able to see what you could maybe even add to the research. And so one of our main things is to incorporate NEFS SC long-term data, which we got from Dr. Paula Fratantoni. So this data covers like a range of years. We only worked with 2019 and 2020, and this data would be included in our temperature and salinity plot. So we would have those multiple cruise paths so that we would be able to see how the water masses have truly changed over a long period of time. And then another thing we could do is separate this cruise track from outbound and inbound. We could also use a higher resolution map because as you can see in our water mass location map, it's kind of just the generic map that R produces, but you are able to read in a script that will give you a higher resolution map. And so overall, we were able to accomplish all of our goals in terms of being able to produce a workflow and being able to compare the 2019 and 2020 data. And so that was very exciting. And we were able to reach our overall kind of like scientific goal, which was reproducibility, which is extremely important, again, in the science community, because you want to be able to have other people reproduce your work in order to make sure that it's actually something. No one ever wants to do research and it's not accurate. So that was very exciting. And so the next steps for me are to conduct research under Dr. Marcia Macedo from the Woods Hole Research Center, and hopefully we'll be doing work on the Amazon this coming school year. I would like to attain my undergraduate degree in civil engineering from Howard University, and I would like to enter a graduate school program in the fall of 2021. And so these are some acknowledgements to my mentor, Dr. Stace Bolu and Joseph Futrell. They have helped me tremendously this summer, not only on learning about water masses and workflows and reproducibility, but also just with specific life skills and steps for moving forward in the future. So I'm very, very, very grateful that I got to work with them this summer. And also a huge shout out to Dr. Fratantoni because she did provide the data for the long-term data that will be used in the next steps of this project. A shout out to LTER because they did fund this project. And then a huge shout out to the PEP program because everyone in this program made this summer possible. I personally don't know what it took to make this summer possible virtually, but I know that all the hard work they did really did make a great summer. So a thank you to them. And that's it.
Thank you, Ayanna, for that presentation. It looks like we have no questions in the chat, so we'll move forward with our next.
Sorry, Joniqua, one just popped up from Heidi Sosik. Great presentation, Ayanna. What would be needed to apply your workflow to another season?
Oh, okay. So, I'm sorry, yeah, you can hear me. To apply the workflow from another season, you would need the data from another season, and you would probably have to reset your guess limits for what you think the shelf and cold water would be. Shelf and slope water would be, because as the seasons change, again, the location of the water masses change and kind of the the parameters within the water masses change as well. So you would need to, we specifically worked with summer, so I know about the summer limits, but you would need to use some knowledge based on the other seasons to input those limits, and then hopefully you will get the same readings as well.
Thank you, Ayanna. Our next presentation will be Exploring the Influence of Helical Swimming and Turbulence of Larval Dispersal, presented by Jalen Dorsett from Delaware State University. Her research mentor is Dr. Lauren Molyneux from HUI. Without further ado, Jalen.
Hello, everyone. I hope you can hear me well. My name is Jalen Dorsett. I go to Delaware State University. And this summer for my PEP summer project, I worked with Dr. Michelle D. Benedetto and Dr. Lauren Molyneux, exploring the influence of helical swimming and turbulence on larval dispersal. So why do we care about larval dispersal? Well, first of all, the larvae of benthic invertebrates are transported based on certain conditions in the water column, such as turbulence, as well as their swimming behaviors. Larval transport aids in learning more about larval dispersal, which is the cycle of them being shot out from their spawning site up into the water column, where they develop enough to where they can come back down and settle. Successful dispersal is very essential for successful settlement, and that helps to make sure that they maintain their populations and avoid extinction. So for this project, we focused on larvae, which are, of course, the immature forms of an organism. And we're interested in looking at larvae that helically swim in the ocean. And by helical swimming, I mean a combination of straight line motion and a circular motion and almost a corkscrew-like trajectory. So you can think of that movement as a slinky toy or something that looks like a double helix for DNA. So for this project, we looked at the way that helical swimming patterns combined with small-scale turbulence can affect overall larval dispersal. And two examples of species that swim helically are the eastern oyster, as well as the marine snail. You can see pictures of them below. So realistically, larvae don't swim in a perfect double helix form. They swim almost side to side looking, so you can see, imagine drawing a squiggly line vertically. These are examples of larvae that were caught swimming helically in a 2D projection. And this was taken from Dr. Stace Below's research. She's also another scientist at HUI. So for this project, of course, we weren't able to get into the lab because of everything going on. So we focused on modeling helical swimming in MATLAB. So everything has been simulated in a 2D form. And you can see on the figure to the left, this was our idealized helical swimming pattern. So of course it looks perfect, but again, this is not exactly how larvae would swim in real life. So we use the equations for dx of dt, which explains our side-to-side swimming component, and dz of dt, which explains our vertical swimming component. Now, again, this is a simulated model, so we looked at different simulations of idealized small-scale turbulence, and we found this example, and of course, U of F and W of F are our horizontal and vertical flow speeds, and they are modeled by these equations. So for the project, Dr. Michelle D. Benedetto made codes in MATLAB to basically simulate our turbulence as well as our swimming behaviors for our modeled larvae. And we use those to show the effects of changing swimming parameters in on-particle spread. The parameters and the codes that we varied were W sub S, which explained our relative swimming, vertical swimming component, U sub S, which was our relative side-to-side swimming component, and SW, which was our relative helical swimming frequency. Now, in the simulation, we modeled about 100 swimmers, but we first tried to look at just how the certain swimming behaviors mixed with either the flow or not the flow would affect larval transport in the water column. So our blue line shows helical swimming, which is a combination, of course, of our vertical and side-to-side motion and no flow. So you can see almost a perfect sinusoidal path that the swimmer would take. The no swimming in flow is our red line. And you can see at the bottom left corner in our circular motion, which can also be called an eddy. They are trapped in that eddy. They can't really move anywhere. They're just going all stuck in that circle. Our yellow line shows our vertical only component in the flow, which is our W sub S. And we can see that the swimmer is able to remove themselves from the bottom eddies and almost transport themselves in a perfect motion. Our purple line shows our side-to-side only, which is our U sub S in the flow. And you can see how they move around horizontally, but they're not able to move up in the water column that efficiently. And lastly, our green line shows helical swimming in flow. So this models almost a more realistic path that the swimmer would take. So when we looked at focusing on our data, we tried to look mainly at our standard deviations of swimmer displacements. So we looked at how much the swimmers would deviate from a center path. So you can see that if the data exhibited a high standard deviation, there would be a lot of spread in the vertical and horizontal directions. So that basically shows that our swimmers are not moving as efficiently. They're not able to swim uniformly together. However, when you look at low standard deviation, you can see there's less spread. So there's less changing in direction of all of the swimmers combined together. So they're moving very efficient and uniformly in a center path. And in our model, we looked at how relative swimming speeds were compared to a flow. So we kind of just said that we were going to set our flow, which was our U of F and W of F, which is our horizontal and vertical flow speeds, to a value of 1. So we compared the swimming, which was our U sub S and W sub S speeds, to that value of 1. So if we increased it over 1, how would that look for our trajectories? So on this figure, you can see the trajectories of many swimmers. And you can see how a lot of them in the vertical direction, which is our Z direction, get stuck below the mean value of 30 units. So basically that's showing how a lot of them, since they are swimming at the same rate of the flow, a lot of them do get stuck in these bottom eddies, so they are not swimming that efficiently. And of course, in our X direction, which is our horizontal direction, you can see that there is a lot of spread. So they're not all moving in the center, which would be our 0 value. So we looked at increasing whichever swimming parameters would make sense to understand how increasing that would change their trajectories relative to the flow. So we kept our flow values at U of F and W of F at 1. So we compared our original trajectories with everything with the swimming and the flow speeds being equal to increasing our W of S over the value of 1. So our vertical swimming patterns were faster than the flow, and we kept the side-to-side swimming the same. So you can see this trajectory shows that a lot of the swimmers are able to stay away from being stuck at these bottom eddies and move more efficiently through the small scale turbulence. A lot of them do deviate from that, of course, that 0 value in the X direction, that could just be from depending on where they start out or depending on just how the flow is. So again, looking at our original trajectory of keeping the flow and the swimming speeds the same, we increased the side-to-side swimming relative to the flow. So that was our U sub S. So we increased that past 1 and kept our W sub S the same at 1, which was equal to the flow. And we saw that there was not as much vertical spread. A lot of them were able to not get stuck in those bottom eddies, and that could be from just not getting stuck in that downward circular path. Might be able to move more efficiently through that so that they are not influenced by the flow as much. Finally, we looked at our standard deviations of vertical and horizontal spread. So we compared an increasing or decreasing standard deviation to an increasing swimming parameter of U sub S, which is our side-to-side swimming, and W sub S, which is our vertical. So when we increase side-to-side swimming over a period of speeds, we saw that increasing side-to-side swimming would actually increase our X spread, so our horizontal spread. So it seems like from where they start off to where their final displacement is, a lot of them end up spreading in that horizontal direction. So of course that can be attributed to, of course, increasing any type of horizontal movement to increasing where they might end up. And when you compare this standard deviation graph to the past, to our past trajectory graphs, you can see that since a lot of them are able to get up from those eddies, the standard deviation, so the spread in the Z direction or our vertical direction, decreases. So they are swimming more efficiently as they increase their side-to-side swimming. And when you increase vertical swimming, there is a peak before you move our swimming speeds past the value of 1, which is our flow. And that just shows that when you increase vertical swimming, of course, there's going to be vertical spread, because without it, you wouldn't have any vertical spread. But as you increase it more, the vertical spread decreases, which shows that the swimmers are moving efficiently together. So a lot of them will not get left behind. And when they are swimming vertically, they're not getting influenced by the side-to-side motion as much. So their horizontal spread will decrease. So I say all this to say that larval transport modeling is very important for parameterizing larval dispersal models for other researchers as well as us in the future. And personally in the future, I would like to look at how larval swimming can impact successful settlement, as an increase in positive dispersal can help to limit, at least from what I've seen in our literature, limit extinction and increase positive influence. The larval settlement process for certain ecosystem engineer populations is important to me, just because without successful dispersal, how can you really have a good population of ecosystem engineers? And they are basically the structure of their ecosystem, depending on what they are. So I think it's important to see if human interaction or human intervention is important and if it's needed. So I would like to thank Dr. Lauren Molyneux and Dr. Michelle D. Benedetto for helping me through this process and going through literature reviews to help me understand the background of this project. I'd like to thank the PEP program coordinators and our mentors for setting up this whole program, as well as guiding us through different career opportunities that we might. I'd like to thank our larval ecology lab group for our literature reviews, as well as just opening my mind to different opportunities. And I would, of course, like to thank the Woods Hole Oceanographic Institution for allowing us to have this program. Thank you.
Thank you, Jalen. At this time, I don't see any questions. Again, I'll just move forward. If you guys do have questions for Jalen, just message her in our chat box here. Our next talk will be determining the presence of haddock within the Stellwagen Bank National Marine Sanctuary using passive acoustic monitoring, presented by Kristen McCauley from Savannah State University, and her research mentor was Dr. Jen Davis from the Northeast Fisheries Science Center. Without further ado, Kristen.
Thank you. Hello, everyone. My name is Kristen McCauley, and my mentor this summer was Genevieve Davis, who works in the Protected Species branch in NOAA, and our research was on using passive acoustics to determine hourly presence of haddock. Haddock, Melanogrammus, Aeglophenis are a part of the Gattidae family, which include cod. They are demersal species, which means they stay on or near the benthos, and they feed on fish and invertebrates. Haddock are seasonal migrators that move to shallow waters during the summer and deeper waters in the winter. They are known to use the mating procedure that is called lekking, which is a special kind of polygynous mating system that includes having an aggregation of males that compete and occupy small territories and then display themselves to attract mates. Haddock have a strong fidelity to spawning grounds, and this causes there to be many stocks of haddock that live within the same space. Stocks refer to the same species, but are isolated geographically or temporally during reproduction. This makes them vulnerable to disruption and fishing pressures. The stocks are managed by NOAA, and Georges Bank and the Gulf of Maine are two of the stocks that they manage in the North Atlantic. The spawning season for haddock usually begins in early February and ends in April, but it depends on location of haddock. The males display a different behavior during reproduction that generally starts with the male occupying a particular space and then performing a dance, which is called the solitary display, or SD for short. At the top right picture, there's going to be a few pictures of the mating procedure, and A is the solitary dance. The fish begins swimming on the floor and swimming in tight figure eights, and it's always accompanied by a regularly spaced knock, which is B. Once a female has entered in the male's area, then the male would display the lead and follow phase, where the male follows the female closely anytime that she enters his area, which is C. The courtship would start by the male swimming tight circles around the female and flicking his fins up and down on her while making humming sounds, which is D. If the female liked it, then they would press their ventral surfaces together and release the eggs and sperm, and mating would be complete for them, which is E. Haddock are vocal spawners and use their sonic muscles to hit the swim bladder to create sound. They use striated muscles, which are called drumming muscles, to rapidly contract and hit the swim bladder to create knocks. The knocks are not just the sonic muscles hitting the swim bladder once, but it's actually two short, low-frequency pulses of sound, spaced closely together. The two pulses differ in frequency and in amplitude and are separated by intervals of 10 to 20 milliseconds. The knocks produced by haddock are repeated at intervals of 140 to 50 milliseconds when they are doing their solitary display dance, and then become continuous when near a female, which causes a humming sound. And I actually have an audio of it. So those knocks were going to be the haddock calls. A way to monitor haddock within an area is by using passive acoustic monitoring in the aquatic environment with the use of underwater microphones, which are called hydrophones. These hydrophones detect and monitor biotic and abiotic sounds in the ocean. Passive acoustic monitoring is a cost-effective and non-lethal way of gathering sounds that complement traditional survey methods, such as aerial, visual, trawl, and telemetry. It helps to gather data in conditions that would not be safe or that would limit other sample efforts. Passive acoustics just listens to noise and sounds that are already in the ocean and is different to active acoustics, which sends out sound and records whatever is transmitted back. A slocum glider, which is shown on the left of the screen, is one of the ways of recording sounds in the ocean, along with others that are mounted on buoys or ships. The area of study is the Stellwagen Bank National Marine Sanctuary, or SBNMS for short, and is a federally protected marine sanctuary. It is federally protected because it is a rich and productive fishing ground due to it being an underwater plateau. The steep sides of the plateau cause deep water currents to rise up when they hit the bank, which causes upwelling and brings nutrients and minerals from the benthos. This upwelling allows numerous species of marine mammals, reptiles, birds, fish, and shellfish to thrive from the nutrient-rich waters. Most of the species that enter the sanctuary are only temporary, but most of them produce noise in the form of clicks, songs, knocks, or snaps. Since the sanctuary is a rich, productive fishing ground, especially for groundfish species such as cod, haddock, and flounder, fishermen take advantage of this productive area. Both commercial and recreational fishing are allowed in the sanctuary, but the NOAA fisheries in New England have put some restrictions that put catch limits on certain species or include large, indefinite year-round closures in the Gulf of Maine. A Slocum glider was deployed in the Stellwagen Bank National Marine Sanctuary from January 2020 to April 2020. The Slocum glider can be programmed to sample at certain locations and depths and is used by the Protected Species Branch to record whale sounds. In this case, I'm using the sounds that have already been recorded to determine haddock calls and locations. The top right picture shows the glider tracks within the sanctuary, which is outlined in the light gray. The green dot is where the glider was deployed and started recording, and the red dot is when the glider was picked up and stopped recording. Once I received the data from the glider, I had to upload it to the sound software Raven Pro 2.0, and it would display spectrograms like shown here on the bottom right of the screen. Spectrograms are visual representations of sound with frequency shown on the x-axis and time on the y. Haddock knocks usually last for 1 to 2 seconds and has a maximum frequency of 650 hertz. I would then record the hourly presence or absence of haddock calls that look like the spectrogram shown. Here is a close-up map of the glider tracks within the sanctuary with some dates to show the speed of the glider. The research questions that I had to answer were, where do haddock spawn in the Stellwagen Bank National Marine Sanctuary, and when do they spawn? Also, are there other areas that haddock spawn that have not been previously recorded? For my results, I did not find any haddock or hear any haddock from January 14, 2020 to March 2nd, 2020, but that does not mean that we did not learn anything from this experiment. No data is still data, and this allows us to wonder why we did not hear any haddock. I looked through 1,159 hours or 49 days worth of sound recorded, and it probably took me about 10 to 20 minutes to look through each hour just to make sure what I was hearing or seeing was or was not haddock, especially since there were other fish that made similar calls or looked similar. Here are some spectrograms that would confuse me while looking for the haddock in the Raven Pro 2.0. The one on the right is another somniferous fish that grunts, but it looks similar to an adult haddock sound. In the middle is the actual haddock spectrogram, and the one on the left is a juvenile haddock that I would hear and see regularly while looking through the recordings. They have a knock sound as well, but the frequency is larger than what it should be and usually ranges from 700 to 850 hertz, while the adult haddock only goes to around 650 hertz. This research is important because having different stocks of fish allows there to be more diversity and allows them to have their own unique ways of reproducing within a species. Fishing pressures can cause stocks of fish to become extinct, and those stocks' unique way of reproducing will be lost. Most external spawning fishes create aggregations like the picture on the top left that causes all of the fish to concentrate in a certain area. Looking for aggregations of fish is an easy way for fishermen to catch the most fish possible with the least amount of effort. But within these spawning aggregations are juveniles and fish that are reproducing for the first time. By removing these young fish from the stocks, it can cause less diversity and can create problems with overfishing because now there are less fish to reproduce with. Cod, which is shown on the top right, is in the Gattidae family as well and was overfished from their high site fidelity and their spawning aggregations in the sanctuary. Now it is illegal to fish for cod and to keep it because of the low numbers they have. Since fishing for cod was not regulated, it caused cod stocks to deplete. Now there is a limit for the amount of haddock that can be fished within the sanctuary, which is a person can only catch 12 haddock that are 17 inches or over to ensure that juveniles and young adults are not fished before they have had a chance to spawn. Going back to the results, haddock were previously found in the Stellwagen Bank sanctuary in 2019, So there are many possibilities as to why I could not find any. The haddock could possibly not be calling or spawning when the glider was passing through because of the water could not have been the right temperature, or the haddock could possibly not be there because maybe they were overfished or their numbers were reduced, or they could have found a better spot to spawn. Some people that I would like to thank is Sophie, Tim, Jenny, and the Passive Acoustic Research Group from the NOAA Northeast Fisheries Science Center for doing this amazing work and research and for being so welcoming. I would also like to thank Mark from the Woods Hole Oceanographic Institution for the glider prep and deployment, and Layla from the NOAA National Ocean Service for providing us with the tools and services to complete this research. And finally, I would like to thank the PEP staff and my 2020 PEP cohort for always believing in me and allowing me to be a part of this awesome community. Thank you for listening, and now I will field any questions.
Thank you, Kristen. I think we have one question. Onji?
We do have one question from Bill Waite. He says, that's brilliant, Kristen. About how close does the glider have to be, how close does the glider have to be to hear a haddock?
I think it just depends on how loud the haddock are calling. Usually they get louder whenever a female is around, or if a male is in its territory, because then it's more of like an aggressive knock. But yeah, I think it just really just depends on the fish, how loud the fish is.
Thanks, Kristen. We do have one more question from Marina Martini. Any plans to use machine learning to identify these haddock sounds?
In one of the papers that I was reading, they said that there was a way to use the haddock sounds to identify individuals within there, because they all have their own certain frequency and the amount of time between the two knocks that they produce. So hopefully in the future, either the protected species branch or a different branch can try to determine individual species of haddock to see if they are coming back to the same place.
Excellent. Thank you. There are no further questions. So our next presentation is the examination of Scope 3 greenhouse gases from Woods Hole Research Institution, presented by Andrea Reagan-Scott from the University of North Carolina at Pembroke Research. Her research mentor was Aaron Bryant Esquire from SCA. Without further ado, Andrea.
Good morning, everybody. I am Reagan Scott. I'm an alumna of the University of North Carolina at Pembroke. My mentor, I had the honor of having a great mentor, Miss Erin Bryant. She works at Sea Education Association. And this summer, we looked at reducing greenhouse gas emissions in the Woods Hole community through a commuter analysis of several predominant Woods Hole science institutions. And it's important to look at greenhouse gases because they're the main drivers of climate change, which can negatively affect terrestrial and marine environments alike. And these scientific institutions in Woods Hole are dedicated to learning about and protecting marine environments. So it's important that they do what they can to try to reduce climate change. So why is climate change a big deal? Climate change has the potential to alter the Earth as we know it. We could expect more intense tropical storms. Heat waves and changing rain patterns are likely to alter growing seasons for important food crops. Rain patterns could also cause droughts in some areas and floods in others that have never experienced rain like that before and are unprepared for it. And we could also expect diminishing or completely changed habitats for terrestrial and marine species. Also, with the increased amount of carbon in our atmosphere, our oceans are becoming more acidic, and it's harder for organisms that live in shales to make those shales and be able to maintain them, which will drastically change food webs in marine environments. So it could cause a lot of major problems that no one is eager to deal with. So the way I looked at reducing greenhouse gases this year was through transportation. According to this 2018 study, transportation was the largest contributor to greenhouse gases in the United States, as compared to electricity, industry, agriculture, and commercial and residential uses alike. So this means that it's very important to examine how we commute and what changes we can make to try to reduce these numbers. So one study from a university in Spain tried to tackle this problem. So this commuting analysis at the University of León in Spain found that cars were the predominant form of travel for all commutes over 2 kilometers. And this is especially surprising because 88% of commutes to this university were less than 4 kilometers. So transportation by car and motorcycle made 96% of the greenhouse gas emissions produced by transportation to this university. And universities are usually ideal places to try to encourage low emitting and active forms of transportation, such as walking or biking or even public transportation, because college students and university staff are more likely to utilize these transportation methods already. Some suggestions the university made were walking, biking, and public transportation. And they found that if they could encourage everyone within a 6 kilometer commute to walk or bike to class, that they could save nearly 44% of the greenhouse gases that they're currently emitting. So big strides could be made with just changing the way people commute in very short distances. However, only 6% of the commutes made up nearly half of the greenhouse gas emissions to this university. So the article detailed that some really innovative solutions are needed to try to address the greenhouse gases produced by people on longer journeys. And teleworking may be an option to help combat this. So we all work and go to school, and regular commutes, not telecommutes, are necessary to get us there. We have to do it. So ideally, we could all live within a mile or 2 of our place of school or work and be able to walk or bike there, but that's just not the reality for most people. So an innovative solution could be telework. Teleworking is practical and viable option to lowering greenhouse gases. It takes advantage of technology we already have at our homes, just laptops and Wi-Fi, to learn or work from home. And this completely eliminates the need for a commute. And teleworking just 1 day a week could have a significant impact in the amount of greenhouse gases that we produce. So we sent out a commuting survey to employees at the Sea Education Association, the Woods Hole Research Center, the Marine Biological Laboratory, as well as the Northeast Fisheries Science Center, to get some data on how these employees commute to work. In the survey, we asked questions about how many days they commute to work, how many miles their average commute is, and what is the average gas mileage that their car receives. People responding to the survey were able to select multiple options for how they commute to work, and I counted all of those options equally when calculating their carbon emissions. So if they selected things like carpool or motorcycle, I counted half of the commute days or half of the average miles, because carpooling and riding a motorcycle equate to about half as much greenhouse gases as driving a single passenger car. So I took the number of days each person commuted and multiplied that with the average miles that they commute each day, and entered that along with their gas mileage or their vehicle into the EPA carbon emissions calculator. And I received a carbon output for each person that responded to the survey. I then recalculated this information with a decreased day of commuting to see the impact that if each person telecommuted one day and only commuted 4 days a week, how much impact that could have in terms of saving greenhouse gases. So we found that we could save 46,797 pounds of carbon if each employee teleworked at least 1 day per week. So this is equivalent to the same amount of carbon as burning over 23,000 pounds of coal each year. It's the same amount of energy needed to charge 2,700,000 smartphones. And it's also the same amount of carbon that could be sequestered by 351 tree seedlings over a 10-year period. In our survey, we also asked about teleworking preferences. So there was a notable difference between those interested in teleworking 0 days per week as compared to those interested in teleworking all 5 days per week. So those not interested in telework, on average, had the lowest commute times. So this is a likely indicator that their commutes do not affect their lives a great deal. Whereas those most interested in telecommuting, those for 5 days a week, on average had the longest commute times, and shows that their commute greatly affected their lives. However, the majority of respondents to the survey selected that they would be interested in 2 to 3 days of telework and respectively 3 or 2 days of work in the office. We also asked people about their productivity levels while they were teleworking from home during the COVID pandemic, and 77.4% of people responded that they were just as, if not more, productive while working from home than they are working in the office. So this is just a good indication that people are self-reporting that they're getting their work done. And if you also notice, the amount of people that said they're more productive roughly is equivalent to the people that said they were less productive, so it ends up balancing out. And there's many obvious benefits to teleworking. The main one, the reason we conducted this study, was to reduce carbon emissions. It's the most exciting one to me. But there's also many other benefits worth noting to the employee and employer. Employees can enjoy work flexibility, so if they have a task they need to do earlier in the morning, they could work a little more in the afternoon, and it balances out fine. There would also be less cost to employees for traveling to work, and they wouldn't have to spend as much money on professional clothing. And there's also increased flexibility with childcare. Employers could save money by not having to rent out as much office space, and it also makes them attractive to prospective employees. So put down the keys, pick up the phone, because we're working from home. So this is just to say that teleworking is a viable option, and these institutions in the Woods Hole community have a real opportunity to reduce greenhouse gases through allowing their employees to telework just 1 day a week. And other people might be interested in teleworking 2 or more days a week. But teleworking is effective, efficient, and it most importantly reduces greenhouse gases. I would like to acknowledge my wonderful mentor, Miss Erin Bryant. She checked on us frequently and also gave us a lot of thought-provoking questions that helped to drive our research. I would like to thank Quinn Bustos for being a great research partner and helping me out. Big thanks to Ms. Nicole Cabana and Dr. Patricia Pinto de Silva for helping us send out our survey to these various institutions. I would like to thank Onji. She played a big role in sending out our survey. Andre checked on me every week, making sure everything was going good with my project. I would like to thank him. Kelly was super helpful with our plots and just overall support. And Dan taught us how to make these awesome presentations. I'd also like to thank the PEP program just providing such a wonderful opportunity, and it's amazing that I was able to learn and gain so much from my home. So big thanks for that. I'd also like to thank NOAA, Marine Biological Laboratory, Sea Education Association, as well as the Woods Hole Research Center for contributing and helping me get my research done. Thank you so much. That's all.
Thank you, Reagan. I see a message, and there might be more, but I'll read this one. Very interesting. This is coming from John Farrington. He said, very interesting. However, what about the need for those who do laboratory and field research?
Yes, there was a section at the end of the survey for people to provide comments, and there were a few people that said that teleworking is likely just not an option for them. And those people were also more likely to select, they were interested in 0 days of telework because they know it's not a viable option for how they do their business. So it doesn't work for everybody. It's not a one-size-fits-all solution, but if it can work for people, I think it's important to look into it to help reduce greenhouse gases.
Perfect. Onji, do you see any more questions?
Yes, Reagan, we have a question from Claire. She says, this research is very timely and relevant to the COVID pandemic. Excellent work. Will you be sharing the results with WHOI's COVID task force?
WHOI's COVID task force. I had not thought about that, but I can definitely write that down. Me and my mentor are finishing up a proposal. We're going to send it to C. Since Ms. Erin Bryant works for C, we're going to try to write a proposal for them and detail out how we think teleworking could be viable for C. And I would love to share it with all Woods Hole research institutions if they would be interested.
Perfect. Any more questions?
We do have one more question, Reagan, and since we have some time, a message from Sandy Sutherland. How much fuel do the research ships use compared to car commutes?
Fuel did, we did not ask about research ships. We asked about just commutes from home to work. We didn't look into that. And I think the main reason we didn't look into that is because that's hard to manage. The science needs to be done on the ship, and there's not a good viable way to do that from home. So we didn't really look into that. But great question, and I think if someone picks up my research after this at sea, then they'll be able to look into that.
Any more questions, Onji?
No, that's all I can see. Thanks.
Perfect. Thank you, Reagan. It seems interesting. It would be even more interesting to note the psychological well-being of people working from home these days, especially with children. Nonetheless, going on to our next presentation, we have two more remaining. It's detecting and mapping abrupt landscape change in permafrost regions of Siberia, presented by Tiffany Winshull from Oklahoma State University. Her research mentor was Dr. Sue Natali from the WHRC. Without further ado, I present to you Tiffany.
Okay, hello everyone. So the project I worked on this summer was detecting and mapping abrupt landscape change in permafrost regions of Siberia. I worked on this project with Sue Natali, Greg Fisk, and Scott Zolkos of the Woods Hole Research Center. So the image you're looking at right now is of a gas emission crater, and I actually never heard about these prior to this project, so it was really exciting being able to learn more about them the past few weeks. So a little bit of background information for this area. The Arctic is actually warming 2 times faster than anywhere else in the world. And as a result of this, permafrost, or continuously frozen ground, is thawing. So specifically in our study region, as indicated in the yellow on the map on the right, the Yamal and Gaidan peninsulas in western Siberia are undergoing very rapid change. So all of the blue areas on this map indicate areas of permafrost. The darker blue indicates continuous, which just means that it's covering almost the whole landscape there. And the lighter blue portions represent more sporadic permafrost, so it's there, but it's kind of patchy. So our area is continuous, however, it is undergoing rapid change right now. So we focused on 2 major changes in this area, one of them being retrogressive thaw slumps. So these thaw slumps are caused as the permafrost thaws, and as the ice in the ground melts, it's no longer there to support the ground. So the ground just starts kind of collapsing. Let me show you. So if you look like right around here, this is where the ground has collapsed on all of these images. These were ones that I found while validating the landscape. And these may not look very big on the pictures, but like this one, for example, this is around 70 meters by 30 meters. and the second one is around 65 meters by 35 and 60 by around 50. So these are very large and can displace a lot of ground material. So the second change we were focusing on was gas emission craters. So because there's only around 7 to 9 known craters, their cause is not 100% known, but potentially they're due to the microbial decomposition of organic matter that becomes available as the permafrost thaws, and that causes a buildup of gas underneath the surface. So if you take a look at the first image on top, this right here, that is the landscape with this hill or mound on it. So that's where the gas is building up underneath the surface. By 2014, in the second image, you can see that the pressure got too high down there and it kind of just exploded into this big crater. By 2015, the crater had begun to erode and fill back up with ground material and snow and water. And then by 2016, if that snow wasn't there, it would just resemble a lake. And there are actually a lot of these lakes that look like that in this area. So sometimes it can be kind of difficult to determine whether or not a lake has been a crater in the past. They were really lucky to have this imagery, this perfect series of exactly how this crater formed, because that isn't the case for every point. Some points don't have this perfect series. So it was really cool to be able to see that. The graph in the bottom, that is a profile of the elevation change for this point. So if you look here, this is the 2013 pre-eruption elevation, around 28 meters. And then by 2014, when the crater exploded, it's way down here around 8 meters. So that's a really big change. And just like the retrogressive thaw slumps, these features can also displace a lot of ground material. So what is unknown about these features and why are they so important? Well, the focus of our project is to understand the frequency and the spatial distribution of these features. If you take a look at this map on the right, this is our study area, and these white dots indicate some of the known craters. And as you can see, they're not all in one location, they're spread out, they're on 2 different peninsulas, so we want to know where they're happening and how many there are. And why do we want to know this? Well, because both of these features can displace hundreds of cubic meters of ground material, there are many risks involved with them, such as to human infrastructure, human populations, oil and gas drilling, because there is a lot of that going on in this area. And you can imagine what would happen if a massive crater just exploded near some oil and gas infrastructure, as well as they are a source of greenhouse gas, but I will get into that a little bit more later. So for our methods, this portion of the methods was done prior to me joining the project, but basically what they had done was use remotely sensed products of surface reflectance and elevation in a Google Earth engine to detect and map the landscape change. So beginning with the photo on the top left, that is our study area, and it has these red grid cells on it. Each of those cells has their own unique ID number to help us keep track of what we validated and where we're at in the process. If you take a look at the photo on the right, if you were to zoom in on our study area, that is the landscape that you would begin to see. And all of those orange pixels on there, those represent some type of change. Now that could mean elevation change, water change, or vegetation. But the thing is, those pixels don't tell you what type of change. It just says something's going on in this area. So in order to actually go through these cells, these pixels, and validate what's actually going on, we have to manually go in and validate what they've detected. So to put the size of our study area in perspective, the Himalayan Guinan peninsulas are around 327,000 kilometers squared. So that's about 12 times the size of Massachusetts. And the area of change, so the area with those change pixels on them, that's about 19 times the size of Cape Cod. So validating this whole area is going to take some time. So the map below, that just shows a randomly selected set of cells for validation. And we're going to start with those because those represent the 13 major land cover types in this area. So that's what we'll start with first. So this is where I come in. My job this summer was to start validating these pixels. So I would be assigned a few of those red grid cells, and I would begin looking from the top to bottom. I would see every change pixel and kind of see what's going on with them. I have the ability to turn it on and off, so I can kind of see what's going on underneath. And if I found a feature that was worth noting, such as this one, that is a turbid lake with some retrogressive thaw slump with it, I would put a point in it, such as what you're looking at on the right, those blue points, they, all of those blue points are tied to a spreadsheet. And the spreadsheet includes information such as the coordinates, so we can go back and find it later. I can categorize the features that I'm seeing, as well as write some notes in there about what type of change I'm identifying. And then my finished project, or my finished product, would look something like what you're seeing on the right. So a cell with all these blue dots on it indicating changes that I found. So for our results, what we found was that 5.09% of our total study area had those orange pixels on it, indicating change. And then of the disturbed areas that I personally validated using the high resolution imagery, 53% of what I found was retrogressive balsam, so that's quite a lot. Additionally, we detected lakes with characteristics of gas emission craters. However, we really need more imagery to be able to confirm these findings. So these three are some that look promising based on elevation change. However, we just don't know for sure yet. So again, I just want to reiterate the impacts of this type of landscape change. So on a global scale, these two features are a source of greenhouse gas emissions. In fact, according to a recent study, soils in permafrost regions store 60% of the world's soil carbon, which can be released into the atmosphere as the permafrost thaws. However, abrupt thaw events such as gas emission craters and retrogressive balsams are not simulated in models of climate change, so we don't know their full impacts yet. However, it is suspected that they will have a greater impact than what was originally thought. Another thing, they are a risk to infrastructure. So earlier this year, I'm not sure if you heard about it, but there was the Norilsk oil spill not far from our study region that was likely due to permafrost thaw. So the holding tank of oil was sitting on the ground, and as the permafrost thawed in that area, the ground became unstable, which caused some erosion to the tank, and eventually that led to an oil spill. And that kind of brings me into my next point about human health. So these landscape changes can impact human health in ways of impacting drinking water for people out here. Even though it's a remote area, there are people and animals and plants and all kinds of things that rely on this land. So it could impact drinking water, ecosystems, and even sewage and landfills if they were to be displaced by ground material. Another thing is, so as I mentioned earlier, we found a 5% landscape change, and that may not seem like much, but that 5% has the potential to impact a variety of things. So this slide was just of some really cool things that I found along the way of validating those changes. If you take a look at the top left, that is just of two lakes, two different colored lakes that are mixing in the middle. And that was pretty cool to find, as well as the bottom right. That was just a dark lake with this bright green smear in it. And then my favorite finds had to be the three in the middle. So those white circular objects, those are either tents, like teepees, maybe yurts, of some sort of potentially indigenous people of the area. And the most interesting part to me is this right here and this up here are reindeer herds. So that was something really amazing to find using satellite imagery. I didn't think that I would see something like that, so that was really cool to see. So what's next for our project? Well, given the rapid pace of change in the Arctic, much of this area is not easily accessible on the ground. So this satellite-based approach provides new insight into the frequency of these abrupt events that are occurring across these landscapes. And as you can tell, like, they can have global impacts. So as you can see on this map, there's a lot of blue. All this blue indicates permafrost, and all of these areas are at risk of undergoing change if the climate in those areas continue to warm. So what we want to do is, the plan is to use this technology on a much wider scale, a pan-Arctic scale, and be able just to detect change in other areas of the world that has permafrost and could be undergoing change as the climate warms in those areas and melts the permafrost. And lastly, I just wanted to say a massive thank you to Sue, Greg, and Scott for letting me work on this project with you guys. I really learned a lot the past few weeks, and this was something that I just had no idea about before, and now it's something that I'm really excited about and interested in. I also wanted to thank the PEP staff just for working so hard to put on the program this year. I know we couldn't be there in person, but this was just such an amazing experience, and I'm really happy to be a part of it. And thank you all for attending to hear what we've been doing the past few weeks.
Thank you, Tiffany. Do we have any questions, Onji?
Yes, we have one question from Andrew Rose. What kind of additional work, either in the lab or in the field, is required in order to incorporate these abrupt events into global models? And there's a second question. Relatedly, are there similar phenomena that we should also be studying?
So in order to be able to calculate how much these features are emitting, you would have to capture the gas some way. I know sometimes if, like with a crater, they fill up pretty fast with water, so you could go in and detect how much gas is in the water. So we would need to be doing more of that, detecting just how much gas is actually being produced out of these. And then the next question, could you repeat that please, Onji?
Sure, the second part is, relatedly, are there similar phenomena that we should also be studying?
Currently, I'm not aware of others. There's probably more, but as of right now, we just focused on these two, so that's all I really know about at the moment.
Thank you. I don't see any more questions.
Perfect. So without final ado here, we're on our last presentation, then we'll have some closing remarks by Onji. Our next presentation is what strategies can help plastic pollution reduction campaigns engage and interact with their audience online. Presented by Quinn Bustos from New Mexico Institute of Mining and Technology, and her research mentor was Aaron Bryant Esquire. Without further ado, Quinn.
Hi, my name is Quinn Bustos. I'm a technical communication student from New Mexico Institute of Mining and Technology. My mentors are Aaron Bryant and George Lyles, and today I'm going to be talking to you all about what strategies can help plastic pollution reduction campaigns engage and interact with their audience online. So go to any beach and you're probably going to encounter some form of plastic pollution. For example, this picture taken in Panama, the most prominent type of pollution is plastic bottles. And plastic is really concerning because it takes a very long time to break down and it can cause harm to marine organisms as they eat the plastic or become entrapped in it. And the more I looked into this problem, the more I saw how preventable it is with some behavioral changes in society that can immediately reduce people's plastic use. And I'm not the only one. On a survey done on employees at various Woods Hole scientific institutions, 95% of them believe that restaurants should reduce their use of single plastic items. So again, as more and more people have realized this, there have been a lot of campaigns established with the goal of reducing plastic use, and they hope to achieve this through behavioral change. And one way they help to inspire this change is through using the internet and social media to do so. And on these platforms, audience interaction and engagement is very important. So one theory that I read about in a paper by Fernandez et al. Was the 5 doors theory of behavior change. And this theory describes the steps and stages that people go through to reach the sustained adoption of a specific behavior change. And in this context, that behavior change is going to be reducing their plastic use. So first is desirability. People have to actually want to make a change. Second is the creation of an enabling context to promote the behavior change. Then following that is can do, where there's going to be an increase in the person's self-efficacy. And then there's buzz, where people are going to want to talk about their steps towards the behavior change. And lastly, invitation, where people invite their friends, family, and others to participate in the change with them. So hopefully, following all of these, there's going to be a trial of the behavior change. And then if satisfaction occurs afterwards, there's going to be sustained adoption. So with my research, there were 3 questions I wanted to answer. First, is there a particular stage of the 5 doors theory that, when appealed to in content, plastic pollution reduction campaign audience members tend to interact or engage with more? How much are interactive features being used by plastic pollution reduction campaigns? And are there any new and engaging features being used? So together, all 3 of these questions would help me answer one central question, which is what strategies can help plastic pollution reduction campaigns interact and engage with their audience online? So to start answering these questions, I first found many different plastic pollution reduction campaigns. There are definitely a lot out there. And to help narrow it down, I specifically focused on those started by young environmental activists. And I looked at each of their social media accounts, went to the last 15 posts on there, and I categorized each post by the stage of the 5 doors theory that the content in them specifically appealed to. And I did so using guidelines, again, provided by Fernandez et al. And I used likes, comments, posts, shares, retweets, all as measures of audience interaction and engagement. So for the interactive features, I simply went to each of the 5 campaign websites, looked around, tried to find what interactive features were there from a list that I had, and this included things like email newsletter signups, contact forms, feedback forms, things like that. So what I found was that audience members tended to interact and engage the most with posts considered or categorized under the 5 doors theory stage of buzz. But this may only be because buzz had the most, it was the most common post type across all 3 platforms that I looked at. It's represented here by the orange bar. You can see on Facebook, Instagram, and Twitter it had the most. But another thing that I found interesting was desirability, represented by the pink bar. It had less than half of the amount of posts than Buzz did across all 3 platforms. Yet it had the most shares on Facebook and surpassed Buzz, as well as had the most replies on Twitter, although Twitter replies were pretty low in general. But I still found it interesting that this specific stage, desirability, was the one for these 2 measures that audience members interacted and engaged with more than Buzz. I also found that all 5 campaign websites had less than half of the 20 interactive features present that I was looking for. I don't really see this as any website design failure or anything like that, but more missed opportunities for increased audience interaction and engagement. I did find some interesting features as well. I found a game on one of the websites, as well as online pledges on some of them. So what does all this mean? It's possible that plastic pollution reduction campaign audience members tend to interact and engage more with content that appeals to the buzz stage and sometimes the desirability stage of the 5 doors theory. Interactive features are not frequently being used by plastic pollution reduction campaigns on their websites, and there are a few of these features present that can be considered new or atypical. So from my research, I do have some suggested strategies for plastic pollution reduction campaigns. First is to produce more content that appeals to the buzz and desirability stages. Not that the other stages should be neglected, but it might be helpful to focus a little more effort on these 2 stages. Also implement more interactive features on their website, again, taking advantage of those missed opportunities, and also aim to include interactive features that are more novel, so that way audience members are drawn to them and more likely to engage and interact with them. So from my research, I hope to help in plastic pollution reduction campaigns, as well as really any environmental improvement campaigns in general, with their outreach online so that they can continue spreading awareness of important environmental issues and promoting lasting change. So many thanks go to Aaron Bryant and George Lyles, my mentors, Reagan Scott, Jess Donahue, Dr. Kara Lavender Law, Dr. Patricia Pinto de Silva, Nicole Cabana, Onjalé Scott Price, and the entire PEP family. Just thank you all so much for all your help, advice, and support with my research and work. Thank you all for being so kind and awesome, and I'm so grateful. I would also like to thank the various campaigns that I interviewed are included with my study, as well as the past and present funders of the Trash Shouldn't Splash initiative, which includes the NOAA Marine Debris Program, the Woods Hole Foundation, the Falmouth Fund of the Cape Cod Foundation, and Falmouth Water Stewards Skip the Straw Project. Also, thank you to the various organizations that we surveyed. Here are my references. And any questions?
Thank you, Quinn. Onji, do we have any questions?
We do, from John Farrington. Another excellent presentation. Much plastic pollution may be coming from people who do not use the web/electronic communications. How do we reach out to them?
So that's where, yeah, in-person outreach is very important too. This is kind of just focusing on one aspect of plastic pollution reduction campaign outreach, specifically online. But yeah, in-person outreach definitely shouldn't be neglected either.
Excellent. Well, I have to say these were excellent presentations all around. So kudos to everyone, round of applause. Are there any more questions? No?
I do not see any more questions.
Okay, so with that being said, I'm gonna go ahead and give it away to you so you can have the concluding remarks.
Yeah, I do agree. Those were absolutely excellent presentations, and not just thinking about the fact that we have not met any of you students or that you were not able to come here. I mean, thinking about those things, that you all did this work remotely is incredible. And even beyond that, I think the work that you all put in and have done has been excellent, especially for those who did not have any coding experience coming into this program, and now being able to show us the results of your code and explain it very well, I think is also just fantastic. I do want to take a moment to especially thank the Woods Hole Research Center for providing this platform for us to host this symposium today. And we're not done. We are going to be taking a break because no one wants to sit in front of the computer for 4 hours straight. So we're going to take a 2-hour break, and we will welcome you all back at 1 p.m. Eastern time to continue our presentations. So students, if you will stay on for just a moment, but everyone else, we will see you at 1. Thank you. We did get a comment from Julia Westwater that says, fabulous presentations. Thank you all.